Welcome, welcome guys to my YouTube channel. If you are new here, I am Andrew Gomande and I make educational videos on the CAPS curriculum. So in our last video, we were discussing and breaking down DNA, the DNA structure. We looked at the function and the history of its discovery. So if you haven't watched that video and you would like to do so, I would leave a link in the description, and also it should appear above in this screen. Okay, so in today's video, we'll be breaking down DNA replication. Okay, so remember, guys, this is CAPS, so this is our CAPS document. We will refer to this most of the time. So we've already covered the location of DNA, discovery of DNA, structure of DNA, role of DNA, and so of course now we have to look at the replication. We discussed the replication and also the DNA profiling as well. Okay, and then of course in our next video we'll be going through the RNA. Okay, so if you remember well, in the last video we said that the DNA molecule has the double helix shape. Okay, so that is how we describe its structure. And DNA is quite important, guys, because through replication, it's able to do a very important function that we will discuss. So, of course, DNA replication, in definition, is the process during which a DNA molecule makes an exact copy of itself, okay, which is what we call a replica. It's an exact copy of itself. Okay, so you have this DNA. Okay, just make a quick drawing there. So this DNA, through DNA replication, will make an exact same DNA. Okay, so this should be identical. Okay, so it should be identical. So we have one DNA molecule making another DNA molecule. Okay, and they will be identical, right? So that's what we are saying. That is simple, because that is replicating. Okay, that is replication. So that's DNA replication, right? Now, when does this occur, and where does it occur? Well, this occurs during the interphase, okay, of the cell cycle. So for those of you who haven't done meiosis and mitosis, or the cell cycles, interphase is the first cycle. Okay, so during interphase, DNA will replicate, especially during mitosis, the type of cell cycle which is your mitosis. It's very helpful there, because remember, in mitosis you are going to um, produce a replica, okay, of the same cell. Okay, and so this has to have the same DNA inside. Okay, these have to have the same genetic material, and so DNA replication makes that possible. Okay, so it divides such that the resultant daughter cells have the same genetic material, or DNA. Okay, so we're saying that it doubles the genetic information so that each cell has the same amount of DNA during mitosis. So this is its significance, or you can say that it is its function. So whenever they ask you the function, this is what you write down. Doubles the genetic information so that each daughter cell has the same amount of DNA. Okay, or the identical DNA, right? Okay, remember mitosis, for an example, when you have, let's say you have bruised your knee, or you get cut. So mitosis occurs such that it multiplies the cells so that it repairs you. Okay, so this wouldn't have occurred properly if you did not have DNA replication. Okay, it's very, very important, such that you replace the cells that were damaged with this exact same cells, okay, the identical cells, right? Um, so of course, DNA replication, guys, occurs inside the nucleus. Okay, in the previous video, again, we said that we have different types of DNA. You find DNA inside the nucleus. You have mitochondrial DNA, and you also have chloroplastic DNA in the animal cell, plant cell. I mean, for the chloroplastic DNA. Right. So this is actually happening. The DNA replication is happening right here in the nucleus. Okay. So let's discuss how it occurs, because a lot of students really struggle to picture all of this. Okay, so we'll start with what happens first, and then of course finish this up. So this is how you describe how DNA replication occurs. Okay, right. So as you can see here, we have a DNA. Okay, so this is a DNA molecule, and of course, it has that double helix shape, two strands, and they are spiral. Okay, that's the double ladder that we were talking about in the previous video. Now, what the first thing that happens is that it untwists. Okay, or what you call unwinds. Okay, so the DNA molecule unwinds such that it goes from this twisted ladder. Okay, that's a bad drawing. Wow. So from this twisted ladder into a side to side strand like that. Okay, straight, not twisted, untwisted. So that's the first thing that happens. It unwinds, guys. Okay, so it unwinds. Okay, unwinds, it untwists. Okay, nothing much there. What else? Okay, well, the second thing that happens is that these hydrogen bonds, okay, remember, these are your nitrogenous bases, and they've bonded. They are bonded. The type of bond is a weak hydrogen bond. Discussed that. So it's a weak hydrogen bond. So this weak hydrogen bond then breaks. That's the second thing that happens. This hydrogen bond breaks, guys. Okay, that's why it is big, so that when DNA replication occurs, it easily breaks. Okay, so of course this breaks, and of course now you have um, two strands that are separate. Okay, they are no longer connected by that bond because it has broken. Okay, so now what happens? Okay, so that's the second part. Okay, we say weak hydrogen bonds between nitrogenous bases break. Okay, and the DNA molecule unzips. So by unzipping, we are basically saying that these hydrogen bonds actually are breaking. Okay, so that is the part, the unzipping part. Okay, the unwinding part, it's when, of course, it untwists. Okay, if that's a word. So it unzips. What do we mean by that? These weak hydrogen bonds break. That's it. That's what unzip means. Now you have these separate two strands, okay, and which are not connected anymore, okay, because you no longer have those weak hydrogen base bonds, okay. Now these two they act as, or they serve as templates. Okay, so they serve as templates such that the free nucleotides that you have in the nucleoplasm. Where is the nucleoplasm? Let's go back there. We said this is the nucleus, right? So, and the nucleoplasm is sort of like the cytoplasm of the nucleus, okay? Because the cytoplasm's job is to suspend these organelles, okay? Remember, all these plasms, the cytoplasm and the nucleoplasm, are both jelly-like substances that suspend organelles, okay? And they are the site where metabolic reactions occur, okay? The reactions that happen inside the cell occur in these, okay? This is not just a space, it's a jelly-like substance. And so inside this nucleus, you have a jelly-like substance which we call a nucleoplasm. Okay. which of course suspends these organelles that are inside, right? So this happens here. Now inside of this nucleoplasm, you have free flowing nitrogenous bases. Okay, your guanine, your cytosine, they are just floating around there. Okay, waiting for a template. Okay, so now that these strands do not have their complementary nitrogenous bases because they have now unzipped, these free floating nucleo nitrogenous bases then bond. Okay, they bond um, um, onto these separate strands. Okay, so you would have, for an example, the thymine. So how many thymines we have? 1, 2, and 3. Okay, we have 3 thymines there. So thymine, of course, will always bond. So it will attach to that adenine, attached to this adenine, attached to that adenine. Okay, so we have used up our thymines. So those 3 thymines have been used up. Okay, so what these are trying to do is to build a new strand. Okay, they are building a new strand by connecting, of course, or bonding, or having their having these nitrogenous bases bonding to this or these original separated um, DNA strands. Okay, so we're done with your thymines, and then of course the thymine will have to bond with the adenine. So how many adenines we have? 3 as well. Okay, because we have how many thymines? 1, 2, 3. Okay, remember, amount of adenines always be equal to the amount of thymines, and the amount of guanines will always be equal to the amount of cytosines. Okay, so these 3 adenines will then bond, of course, with the complementary nitrogenous bases. So you will have adenine here, you have adenine here, you have adenine here. Okay, there we go. So now we are done with adenine. Okay, so we're done with this, and then what else? What is left now? So we are still building up this new strand by connecting, by bonding these free flowing nucleotides. They are nucleotides, by the way, they are not just nitrogenous bases. Okay, so the cytosine will have to bond with, of course, guanine. So where's guanine? There we go. We have guanine there. 2, 3. Okay, because you have how many cytosines? 1, 2, 3. Okay, so these 3 guanines will bond with these 3 cytosines. Okay. Um, Okay, no, that's a mistake. Okay, there we go. Right. So now we get rid of these guanines. Done with that. Okay. All of these nucleotides that were free flowing, free floating there, we have gotten rid of them through bonding with the complementary nitrogenous bases in the original DNA strands. Okay, that's separated. Okay, and then what else? What else is left? Okay, so we have guanine there. So guanine hasn't bonded. So these single strands, they have guanine exposed. So we need to bond that guanine with its complementary nitrogenous base, which is cytosine. So take these cytosines again. They will bond there. Okay, cytosine bond with guanine. There's another cytosine right over here. We bond with guanine. Okay, so there we go. So now you have bonded all the complementary nitrogenous bases together. Okay, such that, guys, such that now you start to form what is what we call um, a new um, a new strand on each side. So this is a new strand. Okay, so they've bonded like that, bonded. So of course, then you have these weak hydrogen bonds forming again. So we form another strand here. These form another backbone of another new strand. So you have two new strands, this one and this one. So these are two new strands. They have bonded with the old strands. Okay. So as you can see now, you have this DNA molecule. It's a DNA molecule, and that's a DNA molecule as well. Okay. And look at this. They are identical, guys. Okay. So they are identical, as you can see. Um, both of these are the same. They are identical, and they look like who? They look like the DNA molecule that we had at the beginning. Okay, right. So and then what happens next? Okay, this results. Okay, so we said that free nucleotides in the nucleoplasm are used to create complementary DNA strand onto each of the original strand. Okay, these are the new complementary DNA strands. Okay, the ones that we have just created now. So this results in two identical DNA molecules. Okay, each molecule consists of one original and one new strand. Okay, so now you have, of course, two identical DNA molecules, and of course they can wind up again. They have already zipped, meaning which these weak hydrogen bonds have formed again, and of course they then form that double helix, which is their natural shape. Okay, so now you have, of course, these two, uh, and of course with those complementary um, nitrogenous bases. Okay, I'm not gonna write all of them, but I just wanted you guys to see how this happens in real time. Okay, I hope I have explained that well to you. That's how you form new DNA molecule. Okay, this is how DNA replication occurs, guys. Okay, you have to picture it. When you picture it, then you understand it. Okay, and so this is quite important. DNA replication, and the significance. Its significance is that it doubles again. It doubles the genetic information so that each cell, which is the daughter cell, has the same amount of DNA during mitosis. Okay, so this means that it results in identical daughter cells during mitosis. Okay, remember in mitosis you are going to the result is two identical cells, guys. Okay, two identical cells, and so of course that means that they have to be the same as their um, parent cell. Okay, so that is the significance of DNA replication. It helps with mitosis, ensuring that each daughter cell has the same DNA, identical DNA with each other and with, of course, their parent cell. Okay, so that is DNA replication, guys. There's nothing much there. As long as you understand the words, guys, use these words, unwinds, weak hydrogen bonds, use those words. Okay, in life sciences, it's very important that you use the terms that we use, or you are going to lose marks. Okay, follow that. Use these terms, and don't rush when you're writing, when you're describing it. Okay, picture it. Okay, go step by step when you're writing it down. Okay, because you may know all of it but get a few marks because you haven't written everything in the way that it's supposed to be written. Okay, so be detailful, ensure that um, you explain as if you're explaining, of course, to someone who as someone who wants to get all the full marks. Okay, So let's move on. So that is DNA replication. Okay. So of course, um, so we're done with that. Okay. I hope I did not rush. So we're done with DNA replication, and so of course we need to um, check out and discuss DNA profiling or DNA fingerprinting, which is quite easy really, and they usually have the same questions. I will show you DNA fingerprinting. They usually have the same questions there. Okay, right. So for DNA profiling, what is a DNA profile? Okay, a DNA profile is an arrangement, okay, of black bars representing the DNA fragments, okay, fragments or pieces of a particular person. Okay, so now each person has their unique arrangement of DNA, and it's only identical twins that have the same exact DNA profile. Okay, so um, this is very useful, guys. We can use it in a lot of instances. For an example, there we go. So they would ask you, they will give you a scenario, and then they would also scenario. They will give you a scenario to match um, DNA profiles and a reason for that. Why do you think is that person who has why why do you think that person is a criminal or whatsoever? And then they ask you the uses of DNA profiling. Okay, so maybe they would give you an activity on or in exams. They will give you an activity where there's a paternity test, and of course we use this DNA profiling in this. So you match the DNA profile of the father and the DNA profile of the son. So if, of course, these black bars do not match, okay, so if the son has black bars that look like this, okay, then the father, of course, cannot be this guy cannot be the father of the child, okay. So they have to match, okay, they have to be in the same um, row. Okay, they have to be in the same row. So you ensure that you take your ruler and you line it up here and see if that match. So if it matches, then it has to be positive. It has to be this guy has to be the father of the child. If not, then it cannot be. So your reasoning is that the DNA profile of the father matches the DNA profile, or the black bars match the DNA profile of the child. So that is your reasoning usually there. So we can use this to trace missing persons, to identify genetic disorders, can use it to establish family relations, identify dead persons or animals, identify crime suspects in forensic investigations. So this is very important. And guys, know the uses. These are free marks. Okay, these are free marks. Know the uses, and of course, know how to match. Okay, and I've already given you the reason. Okay, match well. Okay, so now of course let's take a look at an activity on this. We'll have an activity on DNA profiling and an activity on DNA replication. Okay, so that we can wrap this up, and hopefully you would have benefited from this video. Okay, so let's take a look at this. So this is been adapted from the paper 2, June 2017. Right here we have, of course, DNA profiles. Sorry about that. We have DNA profiles. So the diagram below shows a technique used um, in paternity testing. Okay, So remember, we said that this is, of course, used in paternity testing. Okay, we'll be matching the DNA profiles. Okay, so that means that we are looking for a male. Okay, that is the father of this child. Okay, so what we are focusing on, guys, is the profile of this child. Which profile does it match? Of course, it's not going to be the same. Remember, we said that each person is unique. So it has to be similar or have bars or black bars that are quite similar. Okay, so many bars that match. Okay, so in that way, you are able to say that this male is the father of the child. Okay, so let's see here. Okay, what do we have? So I'll just use this ruler, guys, to sort of match this. Okay, right. So let's zoom in here. So we are focusing, of course, on you would have to focus on these bars. Okay, look at which bar matches with looking at the bars of the child, looking at look at each and every one of them, and look at which male has their bars matching up, or most of their bars matching up with the child. Okay, so, but of course, we haven't arrived there yet. So, the first question says identify the technique shown above. Okay, this is what we call DNA profiling, of course. Okay, so this is DNA profiling, that's the technique. So, DNA profiling, so that's your answer there. And then the second question, which is 2.2, says which male is the biological father of the child? Okay, I was expecting that question, of course. So we match. Okay, so match here. So both of these for both of male 2 and male 3 match for the first bar. Let's look at the second bar. None of them match. Okay, none of them, even meanwhile. The third bar, it's male 1 that matches. Okay, so male 1 matches there. So then these bars, so the fourth bar, male 3 matches. Okay, so male 3 matches. And then the fifth, okay, it's male 2 and male 3 actually. Okay, and look at the last one, it's male 1. Okay, so what decision do we take then from here? Okay, so we can say here that, okay, because male 3 has has most of his bars matching with the child, male 3 is likely the biological father. Okay, so I've already given the mentioned the reason there as well. So say that it's male 3, male 3, and then our answer there, which is 2.3, is his DNA profile, or you can say most bars match uh, with the DNA profile of the child. Okay, profile of the child. Right. And then the last question, which is 2.4, um, says state 2 other uses of this technique. How many do we have? We have a lot, guys. So you can say, so these are the types of questions that they will ask you. So it can be 2 other, so it has to be, can include paternity test. So you can go with trace missing person to identify genetic disorders established. So you can go with any one of these, but 2 of them, okay? And then you get your full mark. So guys, I'm telling you, see this? They always ask this. Okay, they always ask this. Okay, I'm giving you the plug here. They always ask this. Okay, when in DNA profile. So you have to score this, guys. So let's try to write these down. Try to write these down because we really want them written down. So we're saying to trace missing persons and identify genetic orders. Okay, so write those down. Um, so to trace missing persons, okay, and also um, identify biological disorders. Right. Oh, and I made a mistake here in 2.2 male 3, right? So we agree that it is male 3 because its DNA profile matches, okay, with the DNA profile of the child. Or you can say that black bars representing, of course, his DNA fragment fragments matches with the black bars of the child. Okay, so this is how you answer those questions. Okay, so that's it, guys. Let's move on to the activity on DNA replication. Okay, so DNA replication, especially when you have to describe the process of DNA replication, it's worth quite a lot of marks. So that's why I'm saying, guys, that you have to ensure that you really follow all of these. Okay, this is what you're going to write down. Okay, the DNA unwinds, weak hydrogen bonds between nitrogenous bases break, and the DNA molecule unzips. Both DNA strands serve as templates. Three nucleotides in the nucleoplasm are used to create a complementary DNA strand onto each of the original DNA strands, and this therefore results in two identical DNA strands. Each molecule consists of one original and one new strand. Okay, you don't have to write this down, the last part right here, the each part. So, but you have to write down the result. Okay, so that's what you write down, guys, for the description of DNA replication. Okay, and of course the significance of DNA replication for mitosis. We said that DNA replication doubles the genetic material so that each daughter cell receives the same amount of DNA. Okay, or the identical DNA. Okay, so that is the significance of DNA replication. Okay, so this is what we wrote down. Okay, so of course, guys, this is what you'd have to write, and I don't want to waste any time by writing all of this, and so that should be it. So in the next videos, what we'll do is RNA will finish up with the RNA, and then look at past papers as well. Okay, so by the time this is recorded during December, so of course by the time um, January comes, so we'll be just revising. Okay, for 10 months, just revising. That's why I just began this series right now. Okay, so yeah, let me know in the comments how you feel about these lessons. Were they beneficial to you or not, and what could I fix? And we'll see you in the next videos.